Well, thank you, Chair, for the opportunity to stand on this stage this afternoon. And kia ora koutou, and greetings to everyone. I've decided to give my today's talk the title Out of the Squares and Into the Circos, Visualizing Ecological Interactions with Circos. This is one of the works I've been involved in recently, where I collaborated with scientists from Manaaki Whenua Landcare Research's biocontrol and ecology team. And we're basically looking at the interactions between animals, plants, and microorganisms. But I'm going to elaborate on that as we continue. Well, I believe here today, almost everyone in this room has at some point come across a news article discussing the population of our all-important honeybees that seems to be currently on a decline. While this talk is not focusing solely on honeybees, I thought I should express that part because it is one of the reasons for the research activities that are currently taking place, looking at animal-assisted pollination. What is animal-assisted pollination? This is a process, those are some of the things that I was going to mention, but I'm getting ahead of myself here. I was basically going to introduce the study, giving a little bit of insight on the motivation behind the study. And after that, I'll introduce the software tool Circos and mention how it was used in the study, and show just a snippet of some of the results we got. Then after that point, I will draw some conclusions on some of the key findings. I have already mentioned animal-assisted pollination, which is a process where animals facilitate plant reproduction by transferring pollen grains between flowers when they go to or visit flowers to acquire nutrients. This is a quite crucial process for crop production, but at the moment it seems to be facing a threat from a few factors, including climate change, land use conversion, overuse of pesticides, and pollinator diseases. Some research studies have reported that when pollinators visit plants, there is an exchange of microorganisms that takes place between the pollinators, which are animals, and the plants. And the exchange microorganisms that take place during that process can end up forming a mutualistic, commensal, or even a pathogenic relationship with the host or the recipient. But the biggest problem is, even though we know about the relationships that can be formed, there is still not enough information on what specific microorganisms do to specific hosts or recipients. That has prompted scientists from the ecology department, team at Landcare, to embark on conducting a literature review, looking at articles that are peer-reviewed and indexed, but in English at this point in time, and that are written on microorganism, pollinators, and plants interactions. After that, they compiled a comprehensive data set with a list of articles and a list of interactions. It was an extensive project, work or process where they had to go through each one of the papers they came across to compile that data set. But what they also encountered was that there is something I forgot to mention, that when there is an exchange of microorganisms that take place when plants are visited by animals or insects, I mean, not just insects, it can also be baits, they end up forming a three-way interaction. So they had that data set that contained over 200 articles. There was that many articles, and the nature of the interaction is three-way. As a result, it was not going to be easy for them to get an overview of what their data set contains. Then they had to come up with a way of summarizing their data set, and they identified Circos as the tool that could enable them to do that and even visualize the frequency of interactions reported in the articles that make the data set. But what is Circos? Circos is a software tool designed to visualize complex data that traditional plots cannot easily represent. It was initially developed for visualizing genomic features and sequences by bioinformatics scientists. And I should also mention that it utilizes a secular layout, making it perfect for displaying relationships or interactions between objects. Even though Circos was initially designed for visualizing genomic features and sequences by bioinformatics scientists, it later found applications in various study fields. And one of the cases where it was used that I found really interesting is where they used it to analyze speeches that were made by the former US presidential candidates during the Democrats versus Republican presidential candidates debate. And I just want us to take 1 minute and look at the right of the slide. We've got that image that was generated by Circos, visualizing the names mentioned by the presidential candidates, the names of other politicians. What appears on the outside of that ring are the speakers, the presidential candidates that were talking. Then we can see that the rings are also colored in red and blue, representing the Republicans and the Democrats. And inside the ring, we've got lines that are kind of joining the segments of the ring, which indicate the names of politicians being mentioned by the presidential candidates. And the segments of the rings also are subdivided, indicating the number of debates that took place at different points in time, as well as different points in space, many different locations. I'm just going to leave it there and leave politics to the politicians and get back to science. From what we've just seen there, we can see that Circos is excellent for producing high-quality infographics and illustrations with a high data-to-ink ratio, detailed layers, and visually appealing symmetries. It also enables one to control each element in the figure, allowing customization to highlight different focal points and details of for various audiences. Here are some of the key features of the Circos software. It is open software licensed under the general public license, which means you don't have to pay for it. It is developed in Perl and is compatible with any operating system. People who prefer Windows, just like me, have to install strawberry PAL or active state PAL to be able to use Circos. But those who prefer the Unix operating system don't have to do anything, since the latest versions of the Unix operating system come with PAL installed already. It has got a simple installation process, and it uses text configuration files as inputs, including a Circos configuration file that contains paths to some more configuration files. And it operates via the command line interface using that simple command right there, sorry, right there. And it generates images in bitmap and vector formats. Now I'm talking about how it was used in this study. I mentioned that our scientists compiled a comprehensive data set containing articles, and they identified the interactions that were mentioned in the articles and put them in a way where they had the order taxonomic rank of the organism that is involved in the interaction, the family taxonomic rank, and the species taxonomic rank. And unfortunately, Circos cannot read or understand Excel files, since I've already mentioned in the preceding slide that it reads text files. It means the data that had been compiled in the dataset had to be transformed into files that the Circos driving PAL script can understand. And we're just going to look at the right-hand side of the slide. At the top there, we had the Excel workbook that was compiled, which was then transformed into text files that can be read by the Circos driving PL script. And after transforming the files, the dataset into the files, then we're able to employ Circos, but via a Python script, because we wanted to do things dynamically. Then after that, we're able to produce images, which takes place at that point in the image. But we also had a problem because our images did not have legends, so we'll be looking at images but not knowing what we're looking at. Then I had to interface an R script with my Python script to now include just legends to the images. Then just a snippet of the results. That's the first image we generated. I think one thing that's worth mentioning is anything that looks red in that image represents a microorganism. And anything that looks green represents a plant. And anything that looks blue represents a pollinator. And on the outside of the outermost ring, we've got labels, which are the order taxonomic names of the organisms that are involved in the interactions. As we move in from the outer ring, we've got the intermediate ring, or the middle ring, which is now the family taxonomic rank of the organisms that are involved in the interaction. Then the innermost ring is the species taxonomic rank of the organisms that are involved. Then right in the middle, we've got that spaghetti looking like thing, which is the links indicating the interactions that are taking place between the species of the organisms involved in the interaction, or it is an indicator of an interaction. When we looked at it, it looked good, but there was not much information given by it. We had all those interactions. One of the things the scientists were interested in finding out is how many times each interaction appears in the more than 200 articles that they had used to compile a data set. But we could not see that in those links. We even tried to color coordinate the links with the organisms. As you can see, some are red and some are blue and some are green, even though the colors are not exactly the same shades. Then we decided to break things down, which was basically split the results, simplify things, and that was going to reduce the density of the links that we see in the middle of our Circos diagram. I'm going to start by looking at the image on the left-hand side of the slide. We split that into two pairs of two interacting organisms. On the left-hand side there, we've got a microorganism or microorganisms interacting with plants. Then the numbers in the legend is the number of, they are the numbers of interactions contained in the data set. And those range between 1 and 6. And we had to use colors so that we can see, I mean, everybody can see that if I'm looking at red, it means I'm looking at that many interactions. This was basically taking one of the interacting entities and treating it like the starting point in the interaction, and the other two as the endpoints of the, in the interaction. For instance, everything there, in that case, starts from the microorganism and ends in either at the plant or at the pollinator. And the other thing I should have mentioned is we have the order taxonomic rank names of the interacting species on the outside of the outer ring. From this information, the scientists were able to say, since there is information that is missing when it comes to determining what the relationship between each specific, sorry, I'll try to keep it. So they were interested at looking, they're interested in looking at the frequency of the interactions, because if an interaction has been repeated multiple times, it means you stand a better chance of analyzing those interactions and come up with better or meaningful results. But if it has been reported only once, it means there are still opportunities to do more research, but at the same time, the results you'll get are not gonna be that great because there is still a lack of information. Well, the other images are just exactly the same as the first one, but with the point of origin organism having been changed. For instance, on the right-hand side there, we're starting from the pollinator, ending at the plant, and starting at the pollinator, ending at the microorganism. And the last one is also the same. We're starting at the plant, ending at the pollinator, starting at the plant, ending at the microorganism. And finally, what I learned from using the software is that even though it is written in Perl, there is no need for one to understand Perl, but a little bit of understanding the syntax of Perl can come in handy, especially when you have to debug the software when it's not doing what you expect it to do. What can also be useful is being familiar with the concept of directories and files, or navigating directories on the command line interface, and the concept of absolute paths and relative paths. In this study, Circos did facilitate the visualization of a three-way interaction, of three-way interaction frequencies, as I have just shown in the images. And that did help our researchers perform further analyses, including hypothesis testing. And that was the end. Thank you.